like Research Fellowship Award, Young Achiever Award, Tamil Nadu Young Women Scientist Award, Best Teacher Award, and to his laurels. And she has more than 224 publications and 9 patents published, and she had guided more than 20 PhD scholars, and he has research activities on that. And he has a lot of interest on developing. Yeah. Madam has a research interest in stem cells, periodontal medicine, and yoga practice in periodontics. Sorry, sir. I apologize for that. Thank you, sir. And now day is yours, madam. Thank you. Thank you so much.
Thank you very much for your introduction. So, very good morning to everybody. It's indeed my privilege to be here today in this international forum organized by the Society, Indian Society of Dental Research. We have our eminent moderators over here, Dr. Balaji and Dr. Arun. And I'm also very delighted to be here in Kanyakumari because of the reason that when I joined my first college after my post-graduation, that was Raja's Dental College. And I'm very happy that I'm back here, home again. It's just like a second home to me. And it's very nice to see that my students who were there at that point of time, might be 20 years back, they are now senior professors and they are the organizers of this conference. So my best wishes to Raja's Dental College. So the subject which I'm going to talk about is basically the association between the oral diseases and the systemic diseases. We had been working in this subject for past more than 2 decades, and I'll be sharing some of my research experiences with you. So I'll be taking you to the journey of my research over the past 2 decades, decoding the interplay between oral and systemic infections. So we know that the oral cavity is basically the optimum place for any kind of bacterial species to grow, whether it is a common commensals, normal commensals, or it is pathogenic microorganisms. And our mouth, that's why we call it as a microbial. We are basically a microbial man. Wherever we find the bacterial species within us. We are not devoid of any kind of bacteria, means anyhow we are not getting the diseases because we are always balanced with our host immune responses. And as and when the balance shifts either right or either left, then we basically go ahead in any form of diseases, whether it can be an oral disease or it can be a systemic diseases. So when we talk about periodontal disease, we would say that periodontal diseases are the most common chronic bacterial infection of the supporting structures of the tooth, which are predominantly associated with gram-negative microorganisms that exist in the subgingival biofilm. Now, what is the most common etiological factor of periodontal disease? Any idea? UG's most common etiology of periodontal disease? Dental plaque, isn't it? Dental plaque is the most common etiology of periodontal disease. And what is dental plaque? Dental plaque is nothing but it is the microbial biofilm which contains matrix, and this matrix harbors various bacterial species which are adherent to one another, to the interfaces and to the various surfaces within the oral cavity. Okay. Now, if we go back in 1998, we would find that Sokrensky et al., he basically divided the various species of microorganisms into various complexes, and he named them as red, yellow, blue, purple, like that complexes he named them. So why did he group the various microorganisms into the separate complexes? That's why, because these bacteria, they always harbored together. They grew together in a same population. They had got the same quorum sensing. They basically could connect with each other, and wherever any kind of particular periodontal disease occurred, they were found in the same area, in and around same area. For example, a red complex bacteria was found to be in the most deepest periodontitis sites of the periodontal subjects. Now, if we go back to the history, we would find that in the early 20th century, medicine and dentistry were finding the reasons to explain that why people became ablated with wide range of systemic diseases. It was then William Hunter in 1910 who basically coined the term oral sepsis, and he said that there could be some infection which could spread to the other systemic organs of the body. Later on, this oral sepsis was replaced by focal infection, which was given by focal infection theory was given by Frank Billings in 1911. He was a professor in Rush Medical College, Chicago, and he basically described the focal of infection as the circumscribed area through which the various bacterial endotoxins and the bacteria, it can spread through the systemic circulation, it can lodge to the various other organs of the body, and it can affect those systemic organs. So over the years, various facts came into existence which could probably substantiate the role of oral infection and systemic diseases, and this probably evolved a newer term which was known as periodontal medicine. So periodontal medicine is an emerging branch of periodontology focusing on wealth of new data, establishing a strong relationship between periodontal health and disease and systemic health and disease. So at that point of time, this periodontal medicine could be known as an emerging branch. So this definition was given by Offenbacher in 1996. But today if we see periodontology, this periodontal medicine is no longer an emerging branch, rather it is a most established branch of periodontology that is dealing with the gums, and it has something to do with the any kind of systemic diseases you take and you have a connection with the periodontal disease. There's nth number of systemic diseases over here. You name a systemic disease and you will find that there is a connection of these systemic diseases with the periodontal infection. You can find the literature review. You can find, if you go to the past studies, you will find a connection. So out of all these systemic diseases, let us say pneumonia or atherosclerosis, anemia, diabetes, dry mouth, fatigue, fever. So all these, there had been some relation which has been given, in the past studies, that yes, there is some association exists between the gum infection and these systemic diseases. Now, apart from all these systemic diseases, the most common 5 to 6 systemic diseases which has been most studied in the near past were diseases related to cardiovascular system, endocrinal system, reproductive system, that is adverse pregnancy outcome, chronic kidney disease, and obesity. Okay, so obese people were more found to have periodontal infection. So keeping these most common systemic diseases in mind, we thought of researching on these specific 5 systemic diseases. And this also became a part of my postdoctoral research, that is Doctorate of Science, where I basically took these 5 systemic ailments and we tried to find out the periodontal link to these systemic ailments. So we thought of finding it out, identifying the periodontal bacteria, most commonly the gram-negative microorganisms, that is red complex bacteria. We also thought of finding it out, identifying the periodontal viruses and also the various inflammatory proteins. So whenever there is any infection of bacteria and the host, so what happens, host bacterial interaction, certain molecules are released, isn't it? Whether it can be mediators or it can be anti-inflammatory mediators. So anti-inflammatory mediators are lower down, whereas pro-inflammatory mediators, they rise high in the level. So we thought of finding it out, what are the pro-inflammatory markers or mediators which are raised in that particular type of systemic ailment. Let us say coronary heart disease. So these were the established cardiac biomarkers which were identified, and there were also anti-inflammatory markers also which were seen. Also in diabetes, there were few other pro-inflammatory and anti-inflammatory markers. So we basically aimed our research into 3 major perspectives. One is the periodontal bacteria, which is most common. Second is periodontal viruses, and third is inflammatory mediators. It can be either pro-inflammatory mediator or anti-inflammatory mediator. And also when we found out certain findings, we thought of going ahead with the periodontal non-surgical therapy. We did a lot of subgingival irrigation, we did scaling and root planing, and then after that we tried to find out what is the after effect, whether these periodontal mediators come down to the normal level or not. Okay, so that was the hypothesis. So first we thought of finding it out, the association between the periodontitis and the cardiovascular diseases. I started my research with this systemic disease. And to my point, I found that there are some of the most common risk factors of both the diseases, periodontal infection and cardiovascular disease. If you say hypertension, low socioeconomic status, diabetes, alcohol consumption, all are common to both periodontitis also and cardiovascular disease also. Now the point is, apart from all these common factors, risk factors, whether periodontal infection could also be one of the risk factor, that was the hypothesis, that was a question mark. So we did lot of literature survey. We went to the past, we searched so many articles, and we found that yes, periodontal infection can affect the cardiovascular system in 4 possible mechanisms. Number 1, direct possible mechanism. Number 2, indirect. Third, through the genetic predisposition. And 4th, through some of the common risk factors. Okay? So when we talk about the direct and indirect mechanism, we see that the periodontal bacteria has a tendency to get ingress. Now, where is the periodontal bacteria found? Within the periodontal pocket or the gingival sulcus. So the periodontal bacteria has a tendency to get ingress into the systemic circulation through the minute blood vessels, okay, and then they get lodged into the larger arteries, let us say coronary arteries or so. Apart from that, these periodontal bacteria, there are certain bacterial products such as endotoxins, gingipenes, they also are found, or they can trigger some inflammatory response to the coronary wall. Okay, that is another thing. And third is that these pro-inflammatory mediators, they may provoke the liver and they may increase the acute phase reactants are mostly released from the liver, which may further contribute towards the formation of atheroma. So keeping this hypothesis in mind, we thought of identifying 8 periodontal pathogen, most pathogenic microorganisms in the coronary plaque of the patient who is suffering from coronary artery disease, and he is scheduled to undergo coronary artery bypass grafting. Okay, we thought of finding it out, these periodontal bacteria, oral bacteria in the coronary plaque of the patient. And we also thought of correlating the identification, the presence of this bacteria with the severity of periodontal disease. So about 650 patients, they were taken, they were screened, and they were selected from Madras Medical Mission, Chennai. And these patients were both male and female, both patients were selected, and these were the periodontal examinations, what basically we did. These are the common periodontal examination which we all do in our department, periodontal department, whenever any patient is referred to our department, like in proving pocket depth, clinical attachment level, and gingival index, plaque index, and so. So basically we know that how much is the attachment loss and to what category the patient is, like as in the terms of the severity of the periodontal disease. Now once this examination was done and it was recorded, the subgingival plaque was taken from the subgingival, from the deepest periodontal site, deepest periodontitis site of the patients. These patients are suffering from periodontal disease, and these patients are also suffering from coronary artery disease, and they are scheduled to undergo open heart surgery. Okay? So once these plaque samples were collected, the next day when the patient underwent open heart surgery, we took the atherosclerotic plaque samples from the patient. So this is the team of my cardiac surgeon, and by name, my patient name is Mr. Natarajan. Where the surgery is being done, I am also there in the operation theatre. So as we all know, like open heart surgery is a long way process. It takes about 8 to 9 hours, and in some patient we might get the plaque, in some patient we might not get the plaque. So it was a very tedious process. So this is, we can see that the sternum has been dissected. We can see the pumping myocardium over here, and the saphenous vein from the left leg was dissected, and it basically to bypass the diseased coronary artery. Okay, so where the coronary artery was diseased, it was left intact, and the saphenous vein was used to bypass the blood flow. Okay. So now the diseased coronary artery was dissected, and a part of the coronary plaque a chunk of the coronary plaque was collected by the cardiac surgeon and it was handed over to us in a sterile vial. Now we have both the plaque samples from the same patient. We have a coronary plaque sample and also we have the subgingival plaque samples. Now once we got the plaque samples, we want to identify the periodontal bacteria, the oral bacteria. So for this we underwent DNA analysis, that is the PCR was done. Through this DNA analysis was done. The DNA template was prepared. These are all procedures for the PCR, you must be knowing it. PCR reaction was carried out. Gel electrophoresis was done. And it was visualized in a gel dog machine. And we got a wonderful picture. So we have an upper lane and we have a lower lane. Upper lane contains the subgingival plaque samples and the lower lane has the coronary plaque samples, okay, of the same patient. Now presence of these fine bands, it basically depicts that yes, the DNA of that bacteria was present. It means that it confirms the presence of the particular bacteria. Now, how many bacteria did we see? We saw about 8 periodontal bacteria. Okay, apart from the red complex bacteria, we tried to identify Echinella cordens, aggregative bacter, Prevotella nigrescens, Prevotella intermedia. So all these bacteria were noted down, were identified. So these were the bands which we basically got as a part of a result. Now these were the clinical samples and we didn't have stains for it. So how to reconfirm our results? To reconfirm, we had to undergo gene sequencing. So gene sequencing was done by Sanger's method of deoxychain termination and we got a wonderful chromatograph. So this was a reconfirmation of our result that yes, there is the bacterial DNA, oral bacterial DNA in the subgingival plaque and the coronary plaque of the patient. Okay, so this confirmation results was blasted and it was later on submitted to GenBank and we got the accession number. So that was the marked successes of our study. So out of all the bacteria, we found that red complex bacteria, because red complex bacteria is mostly associated with the deeper periodontitis sites. It is basically associated with the chronic periodontitis. So it was found to be at a higher percentage as compared to the other bacterial population, pathogenic bacteria. And this was the first study to isolate the Prevotella nigrescens in the coronary plaque samples at that time. So this study was done in 2004 onwards. And that time, at that point of time, it was the first study in India to be taken up to identify the periodontal bacteria in the coronary plaque sample, and it was cited by the American Academy of Periodontology and various other journals. So keeping this study as a base, we thought of changing our aim from the bacterial identification to the molecules, to the various biomarkers. So we know that microRNAs are a part of biomarker which are basically associated, which are the established biomarker for cardiovascular diseases. Now these microRNA are the family of small non-coding RNA molecules that play an important role in the regulation of gene. So keeping that concept in mind, if this microRNAs can be identified at an early stage, what is a possibility that we can prevent the patient from developing the cardiovascular diseases at the later stage. So keeping that concept in mind, we thought of working on microRNA. So basically we took pro-inflammatory microRNA and anti-inflammatory. 146 was a pro-inflammatory mediator and 126 was an wanted to know what relation it has as the disease progresses, and we saw that the microRNA 146A was, since it was a pro-inflammatory mediator, it was at a much higher level in the coronary artery disease patients with periodontitis as compared to 126, which was a healthy biomarker. Apart from that, we also worked with the other biomarkers. Now, interleukin 1 beta, interleukin 6, all of you know, these are pro-inflammatory mediators, cytokines, isn't it? So these pro-inflammatory mediators were definitely, they were at a higher range in coronary artery disease patients, because cardiac disease is also one of the inflammatory diseases, and over that it was superimposed by the periodontal infection. So the inflammatory levels were raised higher, at a much higher rate. And apart from that, trefoil factors, calprotectin, periostein, vitronectin, these are all established biomarkers which were also found to be at a higher level in periodontitis patients, that is patients having periodontal infection. Now we basically thought of checking it out with some of the genetic expression of certain biomarkers. So for that reason, we took NLRP3 and CRD8. Now this NLRP3 and CRD8, it is basically a mediator. You can just understand like that. It's basically a biomarker, it's a mediator. And there is a certain genes are expressed in the patients who are suffering from coronary artery disease. Okay, so we wanted to find out that whether is there any change in the allele, is there any allele change in these patients when compared to the normal patient. So for that reason, we try to identify this. It'll take some time for. Okay. So we found that we found a change in the allele in NLRP3, okay, expression of the blood samples taken from the cardiovascular disease sample, cardiovascular disease patient as compared to the normal healthy patients with only periodontitis. So these were some of the important findings which we found with respect to these biomarkers. And we published our study in the various research journals. Our study was published in American Journal of Periodontology, Oral Diseases, and few other journals, few other important journals. So this was a marked novelty of our research. Then we thought of working on the periodontal disease and adverse pregnancy outcome. Why did we try to take these adverse pregnancy outcome of the patient? Because most of the women who were entering the department and who were coming for the checkup, they were basically pre-eclamptic, and most of them, they had some or the other form of adverse pregnancy outcomes. So we thought of why not to find out whether there is any possibility of oral infection in these women or not. So in that scenario, we basically thought of finding it out, these three important terms which comes under adverse pregnancy. One is preterm birth, low birth weight, and preeclampsia. But out of these three, since preeclamptic women were higher, they were all coming with a more number. So we thought of concentrating on preeclamptic women. So these women were having hypertension and proteinuria. Now the basic concept, like what is a connection between the oral disease and the adverse pregnancy, is that any kind of infection, any kind of oral infection, whether it is a periodontal infection, rather it can be a dental infection also. Okay, so any kind of dental infection, there is chances that these bacteria can ingress into the small blood vessels, into the, then it may also lead, ultimately it may get ingress into the systemic circulation and get lodged into the placental tissue. Apart from that, it may also provoke the inflammatory response in the liver, which may raise the acute phase reactants. So once these acute phase reactants are released, it may further lead to the inflammation within the uterine wall along with the placental tissue. So as a result, what might happen? They may lead to the total inflammation of the placenta, which may cause the damage of the architecture and which may further lead to preeclampsia. On the other hand, these bacterial invasion can increase the inflammation via TLR4 activation. TLR4 activation is there's a pathway, TLR4 pathway activation. So via that pathway, inflammation is increased, and this may also further lead to the production of prostaglandin E2. Now, what is prostaglandin E2? Everybody knows about it. What is prostaglandin E2? It's basically a pro-inflammatory mediator, isn't it? So this prostaglandin E2, the main role of this prostaglandin E2 in the pregnant woman is that it is basically raised at the end of third trimester. But what happens in periodontal inflammation? The level rise higher much before than when it is required. So it basically raises at the end of the second trimester. So this leads to preterm delivery. So this is basically the concept, okay, between or association between the two infections. So keeping that concept in mind, we thought of isolating the periodontal bacteria, the pro-inflammatory markers, nuclear factor kappa B and toll-like receptor 4, and one anti-inflammatory marker. We thought of taking one anti-inflammatory also, PPR gamma, in the preeclamptic women. Okay, so we took subgingival samples from these women at the end of third trimester before the delivery, 1 or 2 weeks before the delivery, estimated, approximately. And when these women delivered, we took the placental samples from them, and we thought of identifying these parameters. We did a PCR analysis we did RNA isolation, cDNA conversion, and for the expression of the bacteria TLR4 and NF-kappa B, and we found that the periodontal bacteria, that is red complex bacteria, was basically, we focused on red complex bacteria. It was found to be at a very high percentage in preeclamptic women as compared to normal healthy. Apart from that, we also found that TLR4 and nuclear factor kappa B was also at a very higher level than as compared to the normotensive women, okay, normal healthy pregnant women. So what was the idea of taking an anti-inflammatory mediator? We thought of associating the anti-inflammatory mediator with the pro-inflammatory mediator. So we thought of doing the analysis association of the PPR gamma, which is an anti-inflammatory, with the nuclear factor kappa B, which is a pro-inflammatory, and we found a negative association with the odds ratio of minus 0.381. So this was a novelty of our finding. So that was the first time we found that yes, there was a negative association found with these two mediators. So we published it in American Journal of Periodontology, and we patented this finding also later on. It was patented. Likewise, we also worked on periodontal viruses in the pregnant women, in the preeclamptic women, along with MIR 155, which is again a very well-established biomarker of the adverse pregnancy outcome, and we found out a positive correlation of these biomarkers with as compared to the normotensive females. So I think we can say, like, these pregnant women who are having adverse pregnancy outcome, mostly these women were from the rural background. So there is a time that we should need to educate these women to take care of the oral hygiene, because a lot of research says that yes, there is a connection and lot more has to be done as far as the preventive measures are concerned. Then we also took few diabetic patients, and we wanted to know the association of the periodontitis and diabetes. And we know that diabetes and periodontitis, they share two-way relationship, and periodontitis is known as the sixth complication of diabetes mellitus. So we know that the periodontal disease, it releases the pro-inflammatory mediators. Once it is there in the blood circulation, it increases the insulin resistance, which may further increase the blood glucose level. And on the other hand, what happens when in a diabetic patient where there is a hyperglycemia, increase in the blood glucose level, what happens? It basically leads to the defective host response, increased infection susceptibility, which may further lead to the periodontal disease. So it may act as a basically food for the various growth of the various periodontal bacteria as well, isn't it? That's why healing is delayed in these patients, in these diabetic patients. So we thought of finding it out some proteins, because we know that in diabetic patient there is a collagen loss, isn't it? Collagen is destroyed. So what is collagen? Basically nothing, it's an amino acid, it is a protein. So we thought of finding it out what exactly are the levels of these proteins in the diabetic patients with periodontal disease as compared to non-diabetic. Okay, so we focused on three proteomes, that is L-plastin, lipocalin and adiponectin. So L-plastin and lipocalin were the pro-inflammatory proteins, whereas adiponectin was anti-inflammatory. And we tried to find out the association of these proteins in these diabetic patients. We took the subgingival samples and the blood samples from these patients, and we did RNA isolation for the identification of these proteins. PCR was done, and we found that the adiponectin, being an anti-inflammatory protein, was at a much lower level in these patients as compared to the lipocalin and the L-plastin. So they were increased at a much higher rate because they were pro-inflammatory proteinaceous. So these protein can act as a diagnostic biomarker later on, and they can be a good source to identify the chance of occurring these diabetic status in these patients at a much earlier stage, thus leading to some prevention measurements. So this paper, our research was published in Oral Diseases. When we thought of working it out with the obese patients, we did a lot of obesity patients with obesity, and we knew that the obese patients, one of the main thing is that their inflammatory status will always be high. You take any of the obese people, their inflammatory status will be high, whether it is interleukin 1 beta or it is interleukin 6. So what happens that when these patients are also superimposed with the periodontal infection. So this further increases the inflammatory status. So that worsens their systemic health as well as their inflammatory status also. So we thought of identifying the reactive oxygen metabolites along with the red complex bacteria, and we found a positive association in these patients. Also, the last part of our research was the patients with chronic kidney diseases. So while we were doing the research in relation to all these systemic diseases, we found a particular set of patients who were suffering from chronic systemic disease, and these patients had come for the medical camp. They had come for medical camp from the rural areas, and these patients were unaware about the oral hygiene. So we thought of, why don't we find out what is the oral health status in these chronic kidney disease patients. They were under control, they were not under dialysis. So we took the oral health parameters in these patients. Apart from that, we also tried to find out the red complex bacteria, glutathione peroxidase in these patients, and also isolated systolic blood pressure. So these 3 to 4 parameters we tried to identify, and we found that yes, there was a positive association of these chronic kidney disease patients with the oral health status, the periodontal bacteria, and the isolated systolic blood pressure. So this was also one of the novel findings in our research, and this was published in Materials, Biology, and Curious. So all said and done, starting from my journey from cardiovascular disease patients till the chronic kidney disease patients over the past few decades, like more than 20 years, out of my 258 publications in total in the field of periodontics, 100 papers were only in the field of periodontal medicines. Because this was very, very conducive. We have to have a very large sample size, to identify, because a small sample size will not work. So we had to take more than 1,000 patients, about 650 patients or so. And moreover, over that, doing a PCR in a larger population was also very challenging. So we had to overcome all these things, but ultimately it took time, but nevertheless we were successful. So each and every novelty what we were trying to identify, we basically patented all our research work, and today we have 13 patents only with relation to the periodontal medicine out of 16 patents from our department. So since this was a part of my DSc project also, so this was appreciated by Government of Tamil Nadu later on when we had submitted. And it was not only the research which was done, there was basically the need, why we are doing the research, that is very important. So where does the research take us? That is basically the question. Now, after said and done doing all these research and now coming to a fact that yes, there is a possibility of the periodontal infection, association of periodontitis with these 5 most common systemic diseases, I think there is a time that every dentist should have a sufficient knowledge in pertaining the pathway from the oral cavity to the various other organs of the body. So we dentists, I think we cannot call ourselves as only dentists. We are not limited to only oral cavity. There is a link between oral disease and the other systemic diseases. So we have to also look beyond our oral cavity, our oral perspective, to see what exactly is happening in the other systems of the body as well. And these biomarkers, whatever we had identified, and these were the novel biomarkers which we had identified and we had patented also. I think we can use these biomarkers, these molecular proteins, as one of the potential biomarkers as a chairside kit later on. These biomarkers can bring down to a chairside kit where the patients, if in case they are having a periodontal disease but they have not yet developed any kind of systemic ailments, if these biomarkers are higher, at a higher level in them, there is a risk, there is a possibility that they may develop the systemic disease. So there is a time to terminate there and then, okay, so that we can control the systemic ailments at a much early stage. Apart from that, the patients who are already suffering with the systemic diseases, and we found that yes, these levels were higher. So what we can do? We can go ahead with the periodontal treatment. So non-surgical treatment can be done, and then we can again check the level of these biomarkers, what was before and what is after. So they can be used as a therapeutic assessment tool in these patients. So apart from that, as a dentist, I would say that I think collaboration is very, very important. I think moderators will also understand that collaboration is important as far as medical fraternity is concerned, because we are only limited, and anyhow whatever these societies are happening or conferences are happening, it's only limited to the dental fraternity. I think we should provide at large, we should collaborate with the medical practitioners in a way, and we should tell them that how important our oral hygiene is and how it is important to prevent our oral health, oral diseases, and maintain oral health in such a way that we should not go ahead with any kind of systemic ailments. Also, from the patient perspective, I would say that patient education is very important. Every patient who is coming in our department should understand the importance of oral hygiene, should understand what exactly is oral health and how to maintain it. It becomes our duty, not a choice. It should be our duty. Apart from that, enhanced community awareness may be derived from media, from the newspapers, from pamphlets, just to make the rural population aware. Because urban population, they will come to us. But what about the rural population? What about the people who are living in villages? No, we need to basically educate them. And most of the dental practice, and the dental hospitals are less in the rural area. So it becomes our duty as a dental surgeon or as a dentist to reach out to them and to make them, know about the oral hygiene measures. Apart from that, I would say that certain recommendation and policy making, not only to the community fraternity, but also to the government of India should be made. State government, all the state governments, whether it is Tamil Nadu government or any other state, I think we should recommend it, because health policies can be recommended. It can be a little bit modified in a way that every, let us say, a medical hospital should have a dental clinic. Okay, so any patient who is entering the medical hospital should first step into the dental clinic, get the oral hygiene checkup, and then he or she should lead to the medical hospital for any kind of systemic ailment. So this should be made mandatory. So with this, I think I could express whatever my research was for the past these many years, and I could give some of my experiences, my expertise, and share with you. I hope we can make a better dental fraternity with a good oral health in future. Thank you very much.
It was a well-presented ma'am. One of the major things that I would like to say is it's very difficult for us to convince the medical practitioners with regard to the significance of oral infections and its role. And the reason for that is that there is not enough evidence for us. There's not even enough evidence for us to convince them also. So I would like to appreciate your research work and you getting it published.
Thank you.
Because that adds to the evidence that we have with them. And another important area which you focused was on the convincing the patients also.
Yeah, that's very important.
That is another area where we can focus on. We don't need the help of the medical practitioners for that.
Yeah.
If we can convince the patients that gum infection has a potential to cause systemic problems, they would be more than willing to undergo treatment. And I know it's very difficult to do this research. It's very difficult to do research, very costly and difficult, as well as to get it published also. That was what I was telling Dr. Vale, especially in such prestigious journals as the Journal of Periodontology and all, which are highly ranked journals. It's a great achievement, madam.
Thank you so much.
And another thing, you're focusing on one particular area.
Yes.
That is what happens in PG teaching.
Yes.
All the
Yeah,
PGs,
all that is
it's a big mixture. But you have focused on periodontal medicine throughout these years. It's a very big achievement, madam.
Thank you so much.
Thank you so much, ma'am.
That was an excellent eye-opener for all of us.
Thank you.
And the research you have done was tremendous, actually.
Thank you.
And recently, I saw your article in Dinamalar.
Okay.
Everyone were shocked when I showed that. And even we did a project in my daughter's school with will gum disease cause heart attack.
Heart attack.
Even the teachers got shocked.
Yeah.
And the next day, your article came in paper.
Okay.
And they were also like...
Yeah. they came to me. Yeah.
whether we should have a good oral hygiene.
Yes. I think the teaching should be developed from the school.
From the school level.
Yeah, that's better. From the school level, rather than
they should,
grown up and then going to the hospital.
Yes, yes.
If the teachers themselves come into the collaboration with us, and they can.
They have to educate the students.
Yeah.
The students will tell the parents also.
Yes.
sir.
Thank you.
Thank you. And audience, if at all you have any doubts, can ask questions. Yes, sir.
Have a doubt. As you know, like commendable research and
Thank you.
it is very difficult to do
Yeah.
this kind of research.
Thank you.
Like getting plaque from the arteries and then comparing it with the whole part. So what I want to know is that have you got any evidence of Alzheimer's disease in periodontal
Yes, yes, yes. Recently, actually, a lot of articles, if you can go back and see the literature research, a lot of articles have come up now. Research has already been taking place between the Alzheimer's. Rather, one of my students, I was supposed to give her a topic for my MD's topic of the association between the periodontal disease and Alzheimer's disease, because I think a lot of molecules, molecular changes are being happening in the brain when the inflammation increases. So there is a possibility that the people who are having more amount of inflammation, not only oral infection, any kind of infection it can be, they are more prone to forget.
Yeah.
So there is a hypothesis.
Got some data online.
Okay.
And what we've done is we have a national system where patient has a number, and so whatever number the patient has for the dental, they will have for the medical also.
Okay.
So then we can look at the online system and go back if you have periodontitis or you have Alzheimer's code. So that is like very easy to
Yeah, we can find out. Yes,
We've
yes.
done a retrospective study,
Yes.
but we don't have a very large data set there
Okay.
because the population is
Is more,
we
yes.
have not found any conclusive results.
Okay.
Yeah, but this kind of studies we can easily correlate, like retrospectively.
Definitely.
Diabetes and
Yeah,
periodontitis.
even we can, with the small data, we can do some pilot study, and then from there we can take up in large.
I'll get back to you on that.
Yes.
We have just collected the data, but not too much of convincing
Okay.
results yet.
Yes.
But you can work on that more.
Yes, definitely. Thank you very much.
Thank you, madam, and thank you, sir. Now I invite the moderator. Now I invite our moderator, Dr. Arun Sadashivan sir, and V. Balaji sir, to felicitate the speaker of the Jaydeep Mahindra madam with a memento and certificate. Now I invite Dr. Ambali madam, the HOD of PMS Periodontology, to felicitate the moderators. Moving on to the next session. Now I would like to invite the moderator of the session, Dr. Jagjit Singh Daliwal, Associate Professor. Dr. Jagjit Singh S. Daliwal works.
I can read it for the speakers. I will read. Okay, you don't need to read.
So once again, I will invite the moderator. Our moderator, Honorable Dr. Jagjit Singh Daliwal, Associate Professor, sir. Dr. Jagjit Singh Daliwal works at the Institute of Health and Science, Brunei. Dr. Daliwal graduated with a dental degree in 1994 from Karnataka University in India, and the same university also awarded him an MDS in periodontology in 1998. The United Kingdom government awarded him the renowned Commonwealth Scholarship in 2002, which allowed him to pursue an MPhil at Newcastle University in 2017. He graduated with a doctorate in craniofacial health sciences from McGill University in Montreal, Canada. The Indian Councils of Medical Research granted Dr. Dhaliwal a 6-month foreign fellowship for biomedical scientist in 2011 to 2012 at McGill University. He has been actively participating in UBD dental research. In addition to being widely published, Dr. Dhaliwal has been attending and giving guest lectures at numerous national and international conferences. His research focuses on oral implantology, periodontology, oral palliative care, and biofilm decontamination. I invite you, sir, to the dais. Now I invite the second moderator of this session, Dr. Tarulata Shangi, has obtained her postgraduate degree MDS in orthodontics from the BDS from prestigious Babuji Dental College and Hospital Davangere, which is one of the oldest dental institutes in the country. She has completed her PhD from one of the reputed NAC A-graded accredited Mohanlal Sukinda University, Udaipur. She is certified orthodontist of an Indian Orthodontic Society with 2 years of working experience in International College as a guide. She has 2 additional diplomas in health education and creative writing, and she is pursuing 1 more diploma in biostatistics. She also pursuing her second PhD. Dr. Shangi has completed various certification courses on research, scientific articles, writing, reviewing. She has keen interest in research and has more 90 publications in international and national journals. She has presented her works in various national and international platforms as invited guest speaker. She is the editorial board member of Journal of Oral Health and Epidemiology run by Kerman University of Medical Sciences, Iran. She is a reviewer on various international journals. She is the fellow International College of Dentistry and fellow General Academy of Dental Education. She has 18 years of academic teaching experience, and currently she is working as a HOD. Yeah. She is the vice principal and professor in the Department of Orthodontics and Dentofacial Orthopedics at M.R. Ambedkar Dental College and Hospital, Bangalore. Now I give the dais to moderator to invite the speaker.
Okay, thank you, sir, for introducing us. And so I would like to introduce the speaker of the day, Dr. Vinod Krishnan. He is a professor and head, Department of Orthodontics, Sri Sankara Dental College, Varakala, Trivandrum. And so he has done PhD and MDS in orthodontics from University of Kerala. And he also is a member of Royal Surgeons of College of Surgeons, Edinburgh, UK. And he has also edited 2 postgraduate textbooks entitled Biological Mechanisms of Tooth Movement and Integrated Clinical Orthodontics. He is also editor-in-chief of the Journal of World Federation of Orthodontics, peer reviewer for various journals, and he's highly published. And he's also got Young Dentist Researcher Award at Greater New York Dental Meeting, and also the Best Teacher Award from Kerala University of Health Sciences in the year 2018. So welcome, Dr. Vinod Krishnan. The floor is yours.
Thank you very much for the introduction, dear moderators. I think it's overflowing a bit. And very good morning to one and all. Actually, after hearing the lecture of Dr. J.D. Mahindra, and the dentistry is so vast and it has gone beyond oral cavity, even to pregnancy, cardiovascular diseases, and the research was tremendous. And from that envelope, I am inviting you all to the local area where we deal every day in our life. That is the oral cavity. And when I was sitting over there, Dr. Johnson said that this hall is purely meant for pedontology, and why are you sitting over here as an orthodontist? And I thought I will speak something about pedontal ligament itself, which actually is the bread and butter of an orthodontist. And without periodontal ligament, there is no life for an orthodontist. And if we get an ankylosed teeth, it is very difficult to move it even in current era also. So, how will the periodontal ligament behave in various situations is my question over here. And as we all know, orthodontics apply forces, and like drug in medicine, force is in orthodontics. When you apply force, you will get the reaction. And we will all strive to apply optimal orthodontic forces to get the optimal response. And as the force increases, actually the tooth movement also happens. It is well described by Burston and later on by Kirchpers, Jackman and Jap Malta. But are we able to apply forces in an optimal manner on every teeth is a question, because the root surface area differs in each teeth, and if you are applying force for incisor, it will be insufficient for molar, and if you are trying to move molar forward, the force will be very heavy as far as incisor is concerned, because we work on reciprocal anchorage. And there will be inflammatory reaction happening. At the same time, there will be mechanotransduction process, that is mechanosensing, transduction and response. These three are the elements which work, in which the cells of the periodontal ligament senses the signal. It actually transmits the signal from the extracellular surface to the inside the cytoplasm and then to the nucleus, and then there will be a response which is elicited from the cell, which is actually divided into three stages, which we call it as mechanotransduction, and is divided into mechanosensing, transduction and response. And there will be release of various molecules when we apply force in deleterious amounts. So what saves us over here is the cell cycle or cell renewal system, in which the mechanical force loading with orthodontic force lead to periodontal ligament remodeling and differentiation. Without that, I already said that there is no tooth movement happening. And the most abundant cells as far as periodontal ligament is concerned is fibroblast, along with bone cells and odontoblast. And periodontal ligament cells or fibroblasts are capable even of getting differentiated into osteoblast, as evidenced by research in recent times. So what we have done, we have devised one experimental methodology in which we have selected a particular amount of patients in which orthodontic force is applied, and the premolar, which are designated for extraction, is applied with certain amount of force, actually 70 to 120 grams of force, in which the bodily movement is happening, and we mark the buccal surface as compression area and the palatal surface as tension area, and we have extracted the tooth, scraped off the fibroblast or periodontal ligament cells, cultured it and analyzed those before and after force application. The one side we have applied force and other side it is serving as control. So this is the picture of a cell culture, in which you can see in the control area there is vast amount of cells growing up, and in the experimental area there is the cell growth is little diminished. And we need to assess two things. Is there any damage happening to the nucleus as far as fibroblasts are concerned? So for that we have done COMET assay. In COMET assay, we actually assess the morphology of the nucleus, in which in control group you can see that the cells are rounded in shape, and in the experimental group you can see there is elongation of the regions outside the nucleus, which we call it as comets. So with epifluorescent inverted microscope, we can see the characteristic comet-like appearance in the nuclei, and which was not actually visible in control specimens. And we have done the length and calibration of the comet with the help of software. And this is the result, in which you can see, compared to the control group, the experimental group, the comet length is actually doubled. And the doubling of the comet is actually significant as far as statistical analysis is concerned. That means some amount of nuclear damage is happening when you apply orthodontic force. That was the conclusion. So how do cells survive out of this? How do cells survive out of this? Cell has to secrete something in order to survive from this trauma. That is heat shock proteins. Heat shock proteins are evolutionary conserved family of proteins whose expression increases whenever cell is subjected to some kind of metabolic stimuli. One kind of stimuli is orthodontics, or even if the cell is subjected to heat, cold, or any other type of stress, this heat shock proteins will be expressed. And this will be acting as molecular chaperones and that facilitate protein assembly, meaning protein binding, secretion, trafficking, degradation, and regulation of transcription factors and protein kinases so that the cells can survive the trauma. So, increased level of HSPs after several type of stress play a role in cellular homeostasis. So, HSP release is essential for cellular homeostasis. So, we have done Western blot analysis to check whether HSP70 is released. You can see the bands in 2, 3, 7 and 8, and you can see the graph also over here in which 2, 3, 7 and 8, the graph is actually elevated. That means compared to the control group, experimental group in which trauma is supplied or in which orthodontic force is applied, there is particular elevation of HSP70 in order for the cells to survive out of the trauma it is experiencing. So, briefly, all these findings demonstrate that orthodontic force generates some amount of iatrogenic effect in the cells, and the cell or periodontal ligament cells regain from this trauma with the help of release of HSP70. And this is published in International Orthodontics 1 year back. So the next one is cellular senescence. In adult population, we are dealing with more amount of adult population these days. Younger population is actually going away from India. They are in search of newer horizons. They are going to Canada, Europe, and all. So what the patients we have is all adults now, and we have to be prepared to treat adults these days. So we need to assess how the cells in adults behave as far as orthodontic force is concerned. So in cellular senescence, actually there are 3 things which are happening. DNA damage response, that is DDR, telomere shortage, and oncogene activation, all will lead to cell cycle arrest, and these are the senescence features which are shown over here. That is, DDR is DNA damage response. Then what happens inside the cytoplasm? It is in the nucleus, it is DDR and SAFH, and inside the cytoplasm there is increase in beta galactosidase, that is SABJAL. Then there is increase in some molecules, and there is release of senescence associated secretory phenotype. The cell will become senescence associated secretory phenotype in which there is less amount of release of certain molecules. So there will be some morphological changes, there will be metabological changes, there is some oxidative damage happening in all these cells. So we have to take care of all these things while we apply forces to these sort of cells. So the big question over here is whether cellular reaction happens in the same rate as far as adult and adolescents. And we all know that fibroblast renews every 60 days. So 60 days means 2 months. We are applying force continuously. Integrin actin linkage is the first one which we have studied. The actin filaments are actually essential for maintaining the morphology of the cell. If there is reduction in number of actin or reduction in the amount of actin, the cell morphology is disturbed. And it will actually help in functioning fundamental physiological functions of cells such as immune surveillance, signal transduction, endocytosis and exocytosis. And integrins, as we all know, that it is cell addition molecule. It actually connects the extracellular matrix, that is collagen, to inside the cell. And it actually attaches to the focal addition kinase inside, in which there is talin, vinculin, and all those elements are attached. So any trauma which extracellular matrix experiences will get transferred from outside the cell through the cell addition molecule to inside the cell, and the cell is going to react. So how is it going to react? We have compared between adults and adolescents, and you can see that the first graph shows integrin levels, and second graph shows beta actin levels. And the first one, you can see that in the younger patients, in the control group, the integrin levels are high. When the force is applied for 28 days, there is reduction in integrin level. That means there is reduction in the signal transduction process happening. That means cell is trying to protect itself. And in the adult patients, it is the integrin levels are already high in the control group, and it actually getting reduced as far as after force application is concerned. Whereas in beta actin, the cell is trying to maintain its morphology, and whenever there is trauma, there is increase in beta actin levels. So actin levels are going to increase. Cell is trying to maintain its morphology as far as this result is concerned. Then, is there any senescence change actually happens in these cells is the other question. So the second molecule which we have evaluated is beta galactosidase, in which we can see that in the control group there is increase in beta galactosidase as far as adult cells are concerned compared to adolescent. You can see the graph of adolescent is less, and in the adult the beta galactose level is already high. And once force is applied, after 7 days, the beta galactosidase level increases in adults, but not in adolescents. The adolescents is actually, it is decreasing. When you see the graph, it is actually decreasing. While in adults, it actually increases in the 7th day, and then there is a change in the senescence activity. That means cell is rejuvenating with orthodontic forces. Then comes this prostaglandin and interleukin 1 beta. Again, we have looked at 7th day, 14th day and 28th day, in which the prostaglandin levels are increasing in level as far as adults and adolescents are concerned, but in adults the levels are very high compared to adolescents. That means the inflammatory reaction happening in adults is more compared to adolescents. And coming to interleukin 1 beta, the cytokine, as far as inflammation is concerned, it's a much more researched cytokine as far as orthodontics is concerned. Interleukin 1 beta is researched in and out in orthodontics. And in this also, you can see that the adult population is showing a definite increase as far as interleukin 1 beta is concerned, and is significant at all levels. Compared to 7th day, it is significantly high in 14th day. It is even highly significant in 28th day. Then acid phosphatase, we all know this is a marker for osteoclastic activity, and the mRNA elevation pattern for acid phosphatase is actually an indicator that bone resorption activity is happening. So there is continued elevation of acid phosphatase in adults and adolescents. Again, in the case of adults, the levels are much higher in the 7th day and 14th day, and the levels are almost at level when in the 28th day. Then comes alkaline phosphatase level. It is actually an osteoblastic marker where you can see whether there is production of actual bone or not. But over here also, it is actually indicative of the response which we usually see in adolescents. Actually, the alkaline phosphatase level is higher as we go from 7 to 28 days. But in adults, it is actually maintaining a same level without any significant rise. So comparing alkaline phosphatase and acid phosphatase, you can see in acid phosphatase level is very high as far as adults are concerned, and alkaline phosphatase level is very low as far as adults are concerned. So that means bone resorption is much higher in adults and less in adolescents. Then rank L and OPG levels. The mRNA levels of key osteoclastogenic cytokine rank L and its physiologic decoy receptor OPG maintained a very low expression pattern for adult. Is concerned. That is, activation of it might be, we suggest or we assume that it might be a physiological defense mechanism to counteract already the resorption process happened with the help of acid phosphatase in this whole process. Although progressively increasing in expression levels, rank L mRNA never exhibited statistically significant levels as far as 14th and 28th days is concerned, while OPG level showed a significant level as far as adults are concerned. This further emphasized the attempt of the immune system to activate rate-limiting step of bone resorption to keep osteoclastogenic rank L to lower levels and promote bone formation. So, then we have looked into osteocalcin. Osteocalcin is solely secreted by osteoblast. It is pro-osteoblastic or bone building, and bone formative response are higher in adolescents. You can see the blue graphs high. The bone formative cells, bone formative responses are higher in adolescents when compared to adults. This result suggests increased inflammatory response from adult periodontal ligament cells at day 14, leading to more amount of bone resorption than deposition. So all of these indicate that adult cells are more prone for resorption, and we are experiencing it day in and day out, whereas adolescents are much more in a defensive mode. So this is a table which actually summarizes what I've already told. The actual values are displayed over here. Then we have looked into the morphologic changes. Till now we were assessing senescence related secretory phenotype molecules like prostaglandins, interleukins, rankle, OPG, acid phosphatase, alkaline phosphatase, osteocalcin and all. So we actually wanted to see whether there is actual morphologic changes in these cells. So atomic force microscopy facilitate nano-Newton level forces and precisely measures topography, cell elasticity, cell membrane and cell membrane associated structures such as well as cell strain distribution. And along with fluorescent microscopy, when you apply with fluorescent dyes, AFM can provide a much better understanding of the mechanical force induced cellular signaling in real time. So this is the AFM pictures which we have got. You can see in top row it is young cells and bottom row it is adult cells. And you can see the control group, the fibroblast structure is maintained or morphology is maintained. Whereas when you apply force for 28 days, you can see there is agglomeration of particles or cellular activity increase in those areas. This is actually the cell surface extensions observed in these cells, which indicates that the cells are very responsive and conversion into a more active state as far as younger cells are concerned. But when you check into the adult cells, you cannot see cell surface extensions at that level. That means the morphological changes are higher, but the activity is very less. This is the picture from confocal imaging. Showed increased fluorescent uptake. This is control group. These two, A and C are control group, and B and D are experimental group. After 28 days of force application, you can see the increase in fluorescent dye uptake as far as 28-day force application is concerned. And in younger group, you can see actually the agglomeration of these dyes to one area. Indicating that there is increased activity in these cells. Whereas in adult, you can see spread out area that indicates the activity is there, but it is much more in a relaxed state. The last one, which I have to tell you about, is about one anti-aging gene, which is we call it as Klotho gene. Klotho gene is the recognized gene for aging process, and it regulates phosphate homeostasis and activity of members of fibroblast growth factor family. Actually, there are two types of proteins, that is transmembrane Klotho and secreted Klotho. The secreted Klotho protein actually reduces the reactive oxygen species levels, it reduces the insulin levels, reduces the insulin-like growth factor levels, and it actually reduces senescence. So, if Klotho gene is present or if Klotho gene level is increased, we can always say that senescence is decreasing. If Klotho gene levels are less, we can say that senescence is increasing. So this is the Klotho protein level which we got. This is protein level analysis, ELISA test, which elicited increased Klotho protein activity in periodontal ligament cells obtained from younger individuals in compared to adult. In younger periodontal ligament cells, the levels are higher when compared to adult. But to our interest, that after 20 days of force application, the levels went 3 times higher as far as adult cells are concerned. but only twofold increase as far as younger cells are concerned. That means orthodontic force levels or orthodontic force application is actually rejuvenating the cells, reducing the senescence activity, increasing the productive capacity of the cell. So briefly, we have published all these results in American Journal and European Journal. The periodontal ligament cells from adult patients exhibited senescence changes indicated by increased beta-galactosidase activity. The increase in inflammatory response and tendency for more bone resorption is evident by increased prostaglandin E2 levels, interleukin-1 beta, acid phosphatase mRNA expression levels, and controlled bone formation response exhibited by periodontal ligament cells from adolescent patients evident by alkaline phosphatase activity and a balanced Rankel OPG activity. The increase in osteocalcin levels observed in adolescent periodontal ligament cells at day 28 indicate osteoblastic activity trying to maintain bone homeostasis. And AFM nano indentation and fluorescent tagging indicated increased surface morphological changes in younger cells compared to adult cells, and they are observed increase in integrin 1 beta and beta actin mRNA levels in younger patients than compared to adult patients. So, and the level of clothogene was higher in younger patients and adult patients, but after force application, the clothogene level increased twofold in adult patients, while in younger patients there is only one-fold increase. So to conclude, all tissues undergo aging changes, and we expect the same in periodontium and response that periodontal ligament cells exhibit to mechanical stimuli. Aging-related changes in periodontal ligament cells lead to altered response due to suppression of potentially dysfunctional, transformed or aged cells, and this might result in damage of telomeres, highly repetitive DNA structures located at the end of chromosomes, repeated cell division in adult tissues that lack or contain damaged telomerase result in progressive erosion of DNA, reduce shelter and binding, and can lead to increase in senescence changes. So, be extra cautious while you load a compromised periodontium with orthodontic forces. We have successfully treated Surabhi, but it should not be the case in all patients. But what matters to us is a smiling face at the end of treatment, not a crying face. So I studied in Rajas. My BDS was from here. I studied over here in 92 to 97. This is while I studied over here with our beloved chairman, Dr. S.A. Raja. And this is my batch photo. And I am performing some mimicry in the stage. And thank you very much for this wonderful opportunity, Dr. Jacob Raja, and Dr. Johnson, Dr. Alex, and Dr. Aravind for giving me such a wonderful platform to present the research findings. And thank you all for a very patient hearing. Thank you very much.
Thank you very much for a very exhaustive presentation. Was this your PhD project?
No, sir. This was actually MDS dissertation project of three of my students.
Okay. So I was just throughout your presentation, I was just wondering, what is exactly the clinical application of these studies?
Yeah, we usually
so I have been
discuss.
wondering, but in the end you said something about clinical application. That's fine. So maybe you can summarize a little bit more of because we've done so much of work. Like what exactly, if you have to file for a grant, how will you convince the assessors that this is going to be the clinical application and this has not already been found in the literature by other researchers? So how will you convince?
Actually, in the current scenario, the aging related research is getting much more attention. And what we are looking at over here is how aged cells behave with force application. It's the area is not only confined to periodontal ligament, but in all other areas. Fibroblast is present everywhere. So actually we can translate this research into more and more areas. And I got these ideas actually from cardiac fibroblastic research. And we have translated some molecules to periodontal ligament fibroblast actually. So senescence and aging is actually a thrust area as far as a research grant application is concerned.
And I also wonder, how did you get the these tissues, live tissues? How was
As
the
I
obtained?
mentioned in my
Yeah, maybe I missed it. Yeah.
we actually collect, we actually extract premolars, we scrape off the cells, we'll culture it out actually.
Right. Okay. Thank you.
Welcome, sir.
Sir, it was an excellent presentation.
Thank you very much.
We are all very grateful to you to present such kind of an elaborative biomarker-related study, sir.
Thank you very
And since your name is synchronous with biology of tooth movement in India, I would. This is not related to particular topic, but biology of tooth movement, yes.
No
I have a doubt related to Invisalign or a clear aligner right now.
Okay.
What force we are applying is for only 10 days.
Okay.
And how we can apply this result to Invisalign, clear aligner, which is only for the 10 days duration.
Yeah, the answer for this is like, in Invisalign, we, in usual tooth movement, we actually move teeth 1 millimeter per month, right? But in Invisalign, we actually divide the tooth movement into 0.25 millimeter per 7 to 10 days. That means for 1 month, we are actually carrying out 0.75 to 1 millimeter of tooth movement. So these two are same, but it is not a continuous force. Invisalign actually delivers interrupted and intermittent forces. When patients removes it, everything goes back. So
And we are also aware that intermittent forces are not going to be
so effective. Yeah. But we can't help it. The world goes towards that way, no?
And Sir, Burstone's theory and the phases of tooth movement, when you consider initial lag phase, post-lag phase, everything goes into wind when you
Yeah,
check out the clear aligner.
Yeah, we are actually
concept.
working on it, and the paper is coming out as far as this Invisalign thing is concerned. We are actually breaking up the research into each Invisalign spray wear and getting the samples at the 7th day, then again at the immediately after wear, and then 14th day likewise. We are actually coming up with that.
Thank you so much, sir. That would be excellent study, I think.
Thank you.
Sir,
I
audience.
have one more. Sorry
No
for
problem,
asking
no
one
problem,
more
sir, no
question.
problem.
Yeah. So,
Always welcome, sir.
is there anything different that you have found in your studies which is different from already what has not been found previously?
In this one, sir?
Yes.
This is a novel research actually. This is published by us only.
Oh. So it's your research only which has found it has never been found earlier, is it?
No, sir. No, sir.
Oh, great. Okay. That's nice. Okay. So the floor is open to the audience for questions. So no questions? You can ask anything related to orthodontics. He's an orthodontist.
Only orthodontics.
Not if you didn't understand this presentation, it's fine. Even I didn't understand.
Thank you very much, sir. Thank you all.
Thank you, sir. Now I invite the moderator Jagdish Singh, Dalit sir, and Tarulada madam for honoring the keynote speakers. Now I invite Jaideep Mahindra madam to honor the moderators of the session. Moving on to the next session, I invite the moderator, Dr. Rija Mool, who's a professor and HOD of Professor of the Department of Periodontics in PMS Dental College and Science, who has completed his BDS in Rajas Dental College and MDS in Periodontics and Oral Implantology in Annamalai University, currently pursuing PhD in Kerala University of Health Science and various national and international publications to her. Now I invite Ambli Madam as one more moderator of the session, who's a professor and HOD of the PMS Dental College Department of Periodontology. And now I invite Arunima Madam, the keynote speaker of this session, to complete the session.
Good morning, everyone. Respected moderators, esteemed dignitaries, and other ladies and gentlemen. A warm good morning to all. Before I commence my presentation, I would like to send warm greetings from PMS College of Dental Science, and we are the knowledge partner for this prestigious ISDR Conference 2024. And I take a moment to express my gratitude towards my alma mater, founder chairman, Dr. S.A. Raja sir, and the present chairman, Dr. Jacob Raja sir. So coming to the introduction, as you all know, tissue engineering is an evolving field. Years back, a great pioneer research work done by Robert Langer and Joseph Vacanti. Their work on bovine cartilage cells implanted into a biodegradable polymer. And they have implanted this particular experimental research work on the new mouse, and they found out that it took a shape of a ear, and they have implanted into the mouse. And it took nearly a year for this to develop. Like the work was successful, and it creates a lot of debates in the field of tissue engineering science. And this work carries a lot of promises in future. And how come? From hematology to tissue engineering. As you all know, the autogenous platelet concentrates began for science in approximately 1954 with one of the first published articles entitled Evidence of an Antagonist Factor 6 in Platelet-Rich Human Plasma. And basically, the scientific rationale for using this platelet concentrate is that this regenerative tool is capable of releasing supraphysiological doses of growth factors which are required for tissue regeneration. And coming to the milestones of platelet concentrates, this journey began from 1954, and still developments are going on regarding the platelet concentrates. And presently, the generations of platelet concentrates you can categorize into first, second, third, and fourth. Now, a lot of researches are happening in the fourth generation platelet concentrates. And for PGs, the classification of platelet concentrates starting from 2009 by Don et al. And ending up with the 2019 by Chu et al. There are so many classifications regarding platelet concentrates. So coming to the biological effects of platelet concentrates. The platelet concentrates can act as scaffolds. It do have the presence of immune cells. It do have blood cells. It can entrap the cells, and it can provide angio or neoangiogenesis. And my question towards the audience, does platelet-rich fibrin or platelet concentrates fulfill the criteria of tissue engineering triad? What do you think? Yes or no? Not fully. The platelet concentrates or platelet-rich fibrin, if you take, it do have the tissue engineering triad components like scaffolds. It do have cells. It do have signaling molecules, for example, the growth factors. But still, there are many controversies and there are many limitations for this platelet concentrates. The first generation platelet concentrates, the PRP, as you all know, it is quite popularly known. It is very commonly used in the medical sciences and even dentistry. It is used mainly for the hair transplantation, skin rejuvenation, skin lifting, lip repositioning, and orthopedic uses, and even for dentistry in periodontics or minor oral surgical procedures, as well as for implant dentistry. And we have chairside kits for preparing the PRP. And coming to the second generation platelet concentrates, that is PRF. And the main interesting factor for PRF is that you don't need to add an anticoagulant. You don't need an activator like calcium chloride. The preparation is, the biochemical preparation is very easy. And it is a moldable material, living biomaterial. That means you can take up into different forms and shapes, like you can form it in a circular form, you can take it as a plug form, you can extend it to a membrane form, even you can use it as a liquid form, the latest variants of PRF. So just have a close look regarding the differences between PRP and PRF. That is, use of bovine thrombin and calcium chloride is required for PRP preparation, but nowadays bovine thrombin is not used due to the disadvantages of causing thromboembolic events. And sudden fibrin polymerization do occur in PRP, whereas slow natural polymerization occur in PRF. It is a rigid network when you look into the structural wise for PRP, flexible fibrin network for PRF. It's a resistant gel, PRP. It's an elastic gel in PRF. So GNATI introduced the concept of low-speed centrifugation in the year 2018. And when it's like 2013 onwards, the research work started off. And with the introduction, and there goes the introduction of various PRF variants like APRF, APRF plus, TPRF, IPRF, etc. So these are all the variants of PRF. That is, start off with the LPRF, where you can see the difference, where the centrifugation protocol was higher, 3000 RPM for 10 minutes. And the standard PRF also, it was 2700 for RPM for 12 minutes. Later on, in the introduction by Kobayashi and Natty et al., they've introduced the APRF concept, and they have reduced the centrifugation protocol. That means 1500 RPM for 40 minutes. And again, when you look into the IPRF, it is again reduced to 700 RPM. So the recent one is the injectable PRF. Again, injectable PRF, you have different varieties, like you can prepare it with 700 RPM for 4 minutes, and IPRF plus, again, you have variations in the time. So like 700, even there are researchers still not convinced, but again, they are reducing the time from 700 to 400 also. So this is a small work which is done by me itself. We have prepared an aloe vera scaffold in Sri Chitra Institute of Research Lab, whereby which we have prepared a lyophilized aloe vera scaffold, and we have incorporated IPRF liquid into it. And we could find activated PRF in the material. So this is a newer one. This is a device which you can use it for IPRF. When you are using IPRF along with bone graft, you can use this material which is recently patented. It's called sticky bone preparation device. And what are the newer advances in platelet-rich fibrin? You have PRF lysate, you have concentrated PRF, and you have bio-PRF. I'll detail this later. And little bit about a sticky bone, which is very, I think most of the clinicians have used the sticky bone in your clinical practices, and which is very easy. It is like you can collect 5 ml of blood in non-coated tubes, centrifuge it as 2,400 to 2,700 RPM for 2 minutes, and you can obtain the 2 layers, and the superficial layer is AFFG, and this AFG is then extracted with a syringe and mixed with a particulate bone graft, and you wait for polymerization to happen. You can mold it, and you can implant it to the desired defect. And this is something new which is called as platelet-rich fibrin, which is something like, if you, the main problem with the platelet-rich fibrin is that this biodegradability. And you can, they say that this platelet-rich fibrin can be, excuse me, this, the main problem with the scaffold is biodegradability. So what they say is that if you want to do a multiple procedures for a person, if you can harvest the PRF and you can store it, you can lyophilize it, and you can store it in a reduced temperature, like around a minus 80 degree in the deep freeze temperature, and you can crush it and you can use it. This is a recent article published by GNAC. Coming to biobone. What is biobone? Which is being used, this is very interesting factor, which is they have used this centrifugation protocol. I will talk to you in detail. Before that, I just want to introduce the PRF variants, various PRF variants available in market. That is, this is called as biobone. They have used a bio-PRF centrifugation machine where they use a horizontal centrifugation protocol for the preparation of PRF. What they do is like it is they have centrifuged. It is for 700 gram to 700 gram G for 8 minutes, and use a syringe to evacuate 2 ml for ppm from the top by using an 18G needle, and the bottom layer, which is platelet, which is rich in platelet and growth factors, and they heat this for, then they gone for a heating and should last for 10 minutes, called as bioheating, and the test tubes with residual liquid PRF should be kept in the biocool device to prevent clotting. And then you use a syringe to evacuate the remaining 1 to 2 ml concentrated PRF, that is the Buffy-Cot, and they pour it in albumin gel. And the interesting factor for this biobone is that it prolongs the availability, viability of PRF, and it has a usage potential for guided tissue regeneration. And the studies say that it lasts for 4 to 6 months. And as we all know that the problem with the current platelet concentrates, the PRF, is that its biodegradability, it won't last for one month or more, and even the growth factor release is also fast. So this is the first commercially available horizontal centrifuge designed for this PRF, that is the bio-PRF machine by Next Gen Biomaterials. And the 5th generation PRF, if you see, is still under research. What they say is that along with the PRF material, you can incorporate stem cells, you can use it for cell sheet engineering, and there are a lot of researches happening for the 5th generation PRF. So the applications of platelet concentrates, you can see in all the fields of dentistry, it is widely used: endodontics, pedodontics, orthodontics, oral surgery, and perio, etc. And these are some clinical cases where we have tried out this PRF material, where we have used it for grade 3 furcation management, where we have used PRF with the BIOS after root resection, and you can see the pre and the post-op IOPAs. And this you can use, another case which we have done for periapical lesions, where we have used the PRF, the membrane as a scaffold. And even it can be used for phenotype modification therapy, which is called as the microneedling of gingiva. You can increase the thickness of the gingiva by injecting the IPRF. And even you will use the LPRF blocks or PRF blocks for ridge augmentation for immediate implant placement or delayed implant placement procedures. and something called as fibrin-assisted soft tissue promotion, where you can use for root coverage procedure or for gingival augmentation procedures. And coming to the topics is like six levels of platelet concentrates. I gave a overview about the platelet concentrates, which was being, everybody knows about it. What problems with the PRF is that this living biomaterial is one biomaterial. We all know that every biomaterial, when it is gone for marketing purpose, or when we used to purchase the biomaterial, but those biomaterial always will have a levels of researches, like they all must have gone through the basic levels of researches, the preclinical experiments, animal experiments, and then gone for the translational research. Where this biomaterial, the uniqueness of this biomaterial is that most of the studies, if you see, decades back, started off its application directly in the human as well as the animals. Why it is so means it is an autologous living biomaterial. But the problem later on, they come to know that when they have not received the clinical outcome as desired, they started going on with the preclinical researches on this biomaterial. So I just want to highlight few areas where active researches are going on in this PRF. So when the why is clear, the how is easy. So are we better to be evidence-based or evidence-informed? As clinicians, we always need to be evidence-informed. And most of the time, when in college, the people who are associated with academics, always we relay and we teach our students to go for evidence-based practice. So 6 levels of evidence in platelet concentrates. Mainly the 6X hierarchy of evidence-based resources is a useful model for guiding clinical decision making. So it includes, starting from the base level, the single studies, synopsis of the single studies, synthesis, synopsis of the synthesis, summaries, and the systems. So I'm just getting you like the current researches combining all the levels of clinical evidences with the platelet concentrates. So this is just a comparison of the classic randomized control trials, pragmatic RCT, and past and current RCT of PRP and PRF therapy. And still you can see from the table itself, it is like the primary endpoints in classic RCT is still narrow, and past and current PRP RCTs is very much narrow, and pragmatic RCT is still in the middle. And the preparation protocol, that is a very important thing which you have to, I want to highlight, that is preparation protocol for classic RCT is highly standardized, past and current RCT is less standardized, even pragmatic RCT is less standardized. Even a lot of heterogeneity still do exist in the publication of this platelet concentrates. And again, if you see the PRGF, no available RCTs or prospective control studies regards to PRGF, and PRF in the case of peri-implant, no controlled clinical studies on the regeneration of peri-implant defects fully. And again, if you see PRGF, in the case of sinus augmentation or advanced implant surgical procedures, only one split mouth study in 5 patients with a histology, only in 2 patients have been reported. So this is just an overview is regarding the evidence-based review, important systematic reviews. They suggest that a positive impact on PRP, though it is there, but as shown in vivo present, a safe PRP is safe and to be do in the effective procedures, but there are still currently available data are insufficient, and systematic evaluation is limited due to the high heterogeneity in PRP preparation and treatment regime. Though it is said that it is giving us a good clinical outcome, still in the case of clinical evidences, the data is still limited. So do autologous platelet concentrates have a role in intraoral bone regeneration? Exactly. A recent study published by Kurinen et al. In period 2023, he has said that autologous platelet concentrate demonstrated to enhance bone and soft tissue healing in periodontal regenerative procedures, and data for LPRF is most convincing. As you all know, there are many PRF variants, but as compared to all the data like LPRF, APRF, or TPRF, among all the PRF, LPRF stands as a true regenerative material. This is one such systematic review which I have published, where we have compared the variance of PRF, and we have found out that still we have a lot of heterogeneity exists between all the studies, and we found out that IPRF remains, in centrifugation protocol wise, IPRF has got a very good effect, but regenerative potential wise again, LPRF takes a superior position. And as I mentioned, why the heterogeneities exist mostly commonly in all these factors, like the blood collection, the centrifugation protocols, and materials pipetting. So, evidence-based review regarding the PRF. As reported by the American Society of Plastic Surgeons evidence rating scale, they say that the preparation protocol for PRP is, there are many types. There are like a closed spin technique and open technique, and single spin technique and double spin method. So double spin method is the recommended method for preparation of PRF, and the quality of evidence is high and the strength of recommendation is also strong. So you can go ahead with the double spin method for PRP preparation. And as I mentioned, for PRP preparation, you need to have the addition of anticoagulants. Like initially they used botrytis, the bovine thrombin. Nowadays they are not using. The preferred anticoagulant for PRP preparation is the acid citrate dextrose. And the quality of evidence is high and the strength of recommendation is still low. And the recommended parameters for centrifugation are, you should use the relative centrifugation force around 100 to 300 gram for 5 to 10 minutes for the first spin, and 400 to 750 gram for 10 to 70 minutes for the second spin. This is the recommended centrifugation protocol for PRP. So this is just a semi-analysis report that the membranes prepared within 1 minute after the blood draw showed clusters of platelets, leukocytes, and blood cells embedded in the well-organized matrix. Whereas as the time interval increases, they found out that the cells were getting, cells were less, the fibers were getting disintegrated. So it is very important that once you prepare the membrane, you have to immediately place it over the defect. And again, the recent evidences regarding the PRP is that Anito et al., who have done a lot of studies regarding PRGF, and she have compared two preparation protocol, that is PRGF-A and PRGF-B. And she found out that it was said that anticoagulant and the concentration of anticoagulant and 10% calcium chloride is mandatory for the initial studies of PRF. But she have proved in her studies is that the new PRGF protocol, you can as well as you can reduce the anticoagulant dose, reduce the amount of sodium citrate, that is sodium citrate and calcium chloride to around 0.4 ml to 20 microliter, whereby which even that protocol is very much bio-friendly for getting the adequate quantities of platelets in PRP. And again, another approach for making the PRP is that turn down, turn up method for preparation of PRP, where they found out, it was introduced by Mercado et al. In 2022, is a recent article where he found out that you can turn down the vacuum and keep it in the centrifuge and do the first spin and take out and do the second spin. And then they have found out that there was like 4.17 folds increase in the platelet concentration in the PRP using this method. So coming to the most controversial, most interesting aspects and the heart of the preparation of PRP is that the centrifugation protocols, where we follow, and many of the centrifuge what we are using the fixed angle centrifuge, fixed angle centrifugation. But the studies have proven that the fixed angle centrifugation have a lot of disadvantages over the material quality. What they say is that during the fixed angle centrifugation, where in fixed angle centrifugation, where the test tube is kept at 35 to 45 degree angulation, whereas in horizontal centrifugation, the test tube is kept it at a 90-degree angulation. What happens is that when you do the centrifugation, the cells will not be evenly separated in fixed angle. That's what the studies reported by Pinto R. et al. Have proved. This is a very important study, and lot of studies are going on with the centrifugation protocols regards to this aspect. And what they say, the cells are, a lot of cells are getting, going for apoptosis fast with the fixed angle centrifugation. Live cells are very less in fixed angle centrifugation, and even the fibrin quality is also very less. So they have introduced the horizontal centrifugation protocols. And from the picture also you can see the differences between fixed angle and horizontal angle centrifugation. So they have decided to go for the optimization of the platel-rich fibrin. And they said that more than RPM, we have to give importance for RCF. I'll come to it later. So that means the initial researches or initial publication, if you see the initial publications dates back to like 2017, 18 time years, you can see the more of the articles were published or focused, they have mentioned the PRF in RPM and time only. Now even the studies, the international publications, they are not accepting the articles without mentioning the RCF. So either you have to get an FDA approved centrifuge with a horizontal centrifugation protocol, or you have to standardize the centrifuge what we have. So G-force, the angulation is more important than the RPM and the time. This is yet another study which we have published regards to the centrifugation protocol comparing the LPRF and APRF, and this is an AFM analysis report where you can see that in LPRF, as compared to LPRF, the centrifugation protocol is 2700 RPM for 10 minutes and APRF 4400 RPM for 40 minutes. You can see the difference. The fibers which we have obtained in LPRF, which is much more denser, is thick as compared to APRF. The APR, the fibers were very thin, and it is the interfiber distance is also very more. Like what they say that but the cells, we have done the surface microtopographic studies, and again in our studies, again we found out that the histological studies that the cells and the growth factors released were more in APRF as compared to the LPRF. So what you can, what inference you will get is that if you need a material which you need to have to withstand the force, means you have to use a LPRF. But quality-wise, if you go more centrifugation forces, I mean, if you reduce the centrifugation force, means the biological characteristics of the material is superior. But the mechanical properties-wise, you have to go for LPRF. That's why the LPRF use is more commonly done for implant purposes. And for quality-wise, but for any researchers, for regenerative purpose, we need both the properties, right? We need the mechanical properties, and you need the good biological properties in the material. So the Mirren standard, one article which was published by Mirren in 2000, I read it all in 2020, and he said that the optimal centrifugation speed and protocol. This is a good article. Actually, they have said that you can use this standardized protocol between like 400 to 700 for 8 minutes within the investigator ranges. And the protocol for 700 gram for 8 minutes yielded good quality platelets and leukocytes. And they were distributed throughout the PRF layers. This is yet another study published in 2018. They have supported regarding the optimization of the PRF, and they said that use of horizontal centrifugation of PRF is opposed to standard fixed angle devices led to a 4-fold increase in cell separation. And now coming to one interesting aspect is that, as I mentioned before, when we refer the articles, there are a lot of heterogeneity regarding the centrifugation protocols. And it is said that, you can even incorporate this AR2T3 guidelines in your articles or when you're doing any publication. That means you have to give importance to the centrifugation protocols like angle radius, relative centrifugation forces, revolutions per minute, time of centrifugation, tube size, and tube characteristics. All these needs to be standardized. So this is what this AR2T3 protocol, from a publication point of view especially, this is that by changing the angle of centrifugation from 0 to 90 degree is the distance from the rotor to the tube increases, right? From the picture it's clear that a green line running in the central line, and this increases the G-force and its effect over the blood component. So as I mentioned before, in the fixed angle centrifugation, we were using 35 to 40 degree tube angulations. You can tilt it to 90 degrees, 0 to 90 degree. So the relationship between relative centrifugal force and time is that increasing the relative centrifugal force will accelerate the sedimentation of the blood components, and thus does the composition of the PRF. So modifying the time of centrifugation modifies the time of G forces, and thereby it influences the blood composition. So coming to the quality of the piece, the platelet concentrates, you can, I think from the picture itself is very clear that if you delay, like that the prepared material in 30 seconds how it is, 1 minute is what is happening, and 2 minutes the changes in the morphology. And coming to the storage of the platelets, there are many controversies regarding the storage of platelets. There are no long-term studies have been performed except for one 6-month study with equine PRP, where it was reported that PRP can be stored in minus 80 degrees Celsius for 1 month to keep the concentration of the growth factors like TGF beta and insulin growth factor 1 intact. Whereas the study recently by the Mader beta et al. In 2023, this study said that freezing is an acceptable preservation method. It can be done. You can harvest the PRF material and you can store it at minus 20 to minus 80 degree for up to 3 months. So these are all the evidence-based review regarding lot of studies. Actually, it is a very vast topic, lot of articles, and still, but what we conclude is that there is still a lot of controversies, and this is an area for everyone to do research. And they say that one aspect is that the healthy patients show differences in the composition of PRF between men and women, with sex being very great factor in age. And there are controversies regarding that the quantity of the platelets is more in men than women. There are other contradictory studies regards to it. Age is a very important determining factor than sex in the preparation of PRP. That means the systemic diseases can do affect the quality of the platelets. So those people are immunocompromised, the quality of the platelets, activity, the function will also be compromised. So these are the limitations. As you all know from the graph, it is very clear that if you can do a combination like autograft as the gold standard biomaterial for regenerative purpose, you can use autograft plus PRF. It is more realistic and it is ideal. And you can see where the graph goes down, where like if you use a PRF as a solo biomaterial, it is not proving its full efficacy. So the disadvantage of the platelet concentrates that systematic reviews contradict the efficacy of platelet concentrate due to the following reason, as I mentioned before: the fast degradation rate and inability to sustain the long-term growth factor release, and poor mechanical properties and mismatch with the complex regenerative environment. Next, the third one is difficulty in maintaining the long-term structural integrity. And for which, to overcome these drawbacks, now the researchers are going on the scaffold system are needed to protect and stabilize this autologous platelet concentrates, as well as to modulate the growth factor. We need this living biomaterial and we need its full potential, for which they are going to modify the PRF. You can use hydrogel-based APC modification strategies. There are innumerous articles on it. And APC modification using nanoparticles and APC modification using electrospinning. During electrospinning, for example, if you're spinning polycaprolactone material or a PLA, you can even spin the fibrin material along with it. And 3D printing based APC modifications. These are all areas where anybody can do research. And why it is still in research, there is an unstable positioning of PC therapy in regenerative dentistry, is that higher performance bias, an unidentified and complex mechanism of action of platelet concentrates, and meta-analysis have been performed on the data obtained from such poor RCTs varies from cases to cases. and PRP and PRF clinical trials, and it is not observed using clinical trials of it. They have not standardized the clinical trial protocols for this biomaterial, and therefore a poor or irreproducible clinical outcomes based on solo applications. So it is not justifiable if PRF is going to be used as a solo biomaterials. Thus, you can conclude that platelet concentrates therapy in dentistry or regenerative medicine is recognized as an adjuvant therapy. So to conclude, based on the evidences, the science behind the preparation of platelet concentrates is still in its infancy. We are far from producing the ideal platelet concentrates and using this autologous biological product to its full potential. So again, as I want to highlight that standalone or solo PCs application has a low levels of evidence. Combining PC with any kind of biomaterials is therefore a viable synergistic strategy for the present regenerative use in dentistry. So I conclude, I sincerely thank all the organizing committee members of the ISDR conference for giving me this opportunity and immense support. Special gratitude, and I'm so privileged to do my presentation in my alma mater, my beloved teachers, my well-wishers, my friends, my students, my postgraduates, and I wish all the success, and may God bless you all. Thank you.
I congratulate Dr. Arunima for taking up this topic, which touches the actual truth about PRF. Those who are doing publication, they will only realize that we need actual centrifugation, not only centrifugation protocol, the G-force conundrum, and the RPM things. And when we send articles for good journals, it's mandatory that we have to follow certain protocols in these days, as explained by Myron et al. In 2020, 21, and 22. Good protocols are envisaged. As you rightly said, being an autologous biomaterial, and which is cheaper, which we can easily take in our clinical setting, we all jumped into PRF for using it in every defect that we find. But the reality is that unlike most of the other biomaterials, in vitro, the basic research findings are lacking in most of the platelet concentrates. And we are now going back to find out the actual truth about PRF after using it in variety of clinical situations. And we have to do a lot of basic research work that is lacking to find out the actual use of these biomaterials. And that is what she has talked about in this presentation. I really appreciate for taking up this topic, and wish you all the best in your future research.
It was an excellent presentation. Thank you,
Rachel.
I think it will be very useful for postgraduate students for doing more research on this advanced techniques and all. Thank you.
Any questions from the audience? As Johnson mentioned, I think a lot of periodontists are here. Thank you all.
Thank you, madam. Thank you for the innovation. Thank you for the innovative talk and applications of PRF in various aspects of periodontology. And now I invite our moderator, Rijamal madam and Ambli madam, to give the certificate and the mementos to the Arunima madam. No. Now I invite Namita Sharma madam to give the mementos and a certificate to our moderators. Madam, please to join. Arnava madam, please to join. Thank you, madam. I think that's a small technical thing. Now we can break for a tea break and we'll join by 10 minutes. Sorry for the inconvenience. The speaker is preparing their points, so that's we have a difficulty. Madam? The next speaker is Dr. Dana Yark on the topic of aesthetic medicine. Topic is on ecological medicine, artificial intelligence, post-traumatic stress, aesthetic medicine and photobiomodulation. Madam, I have told him to find some people because we had an
Okay,
issue.
good.
So we will manage it.
Yes, we will.
Shall we continue the session? Yeah. Sorry for the break. Now we got all this corrected. Can we continue the session? Yeah. So now I invite Dr. Donna York from USA to give a keynote address on the topic of ecological medicine, artificial intelligence, post-traumatic stress, aesthetic medicine, and photobiomodulation. Thank you. The dais is yours, madam.
Good morning. Namaskaram. It is absolute great honor to be here for Raja College. I'm a local girl. I'm a Kerala girl from 2015, and please give applause to Dr. Abby Alexander, who is my student from NYU. Dr. Abby, please come in front. Have you seen my purse anywhere? My purse, no? Oh, it's here. So Dr. Abby was my student when he was in the second year dental school at NYU. And we are very proud of him of being a very faithful Indian and Kerala member, because he brought his teachers from NYU here, and he became himself a wonderful doctor and a wonderful colleague and very faithful friend. So please applaud Dr. Abby Alexander, who is dedicated to Indian medicine and dentistry. And one important thing is this amazing intellectual capacity of India. Congratulations.
All right, thank you.
In NYU, we have students from all the world, but nobody is more impeccable, serious, attendant to indications as an Indian girl. Boys are not disciplined, but girls they are all the time. So if you want the best student, you must have an Indian girl student. Is that right? So, I am Dana, and why do you think I'm here in front of you? Because I went to school. Do you know how much I went to school? Oh my God. I went to school first for doing piano, then for doing painting. Then I went to school to become a dentist. And I did 6 years of dentistry program, yes? And I did a master in biophysics and cellular technology. And then was not enough. I went to PhD for 7 years. Was that enough? No. I went to 2 years to school, to Academia of Distinguished Educators in New York at NYU. Was that enough? I think was enough. So then now I want to share with you everything that I'm passionate of. Whatever I'm passionate of, I want to share with you. So those are my passions, and I'm gonna be fast. Also, everything we talk about, and other lecturers, brilliant lecturers that we have for EMLA, that means European Medical Laser Association, you find it on our website. There you go. There is no sound. Can you please link the sound? This is the Star Trek. Remember in the Star Trek, it was this man that was having a ship, and on the ship, they were having all kinds of lights. So in the ship, they were treating with light, right? And you can see how she's treating in the film. Do you know Star Trek, the film? You should look, it is on the Netflix and stuff. And at that time, they said, oh, you put a light and it's gonna do the wound healing. And it's true, now we use it all the time. No, doesn't want? It's okay. It's okay, if it doesn't want, it's okay. We don't need it. What is this mercury toxicity? Mercury toxicity is when you get poisoned from mercury, but they didn't know about this mercury toxicity, so they thought it's good for you. And then in Greece, they were using it, the Romans and stuff, and then they use it in the Peru, the Incas. They use it to do fountains. They loved mercury because it's so handsome and beautiful. They did give it to people as drinking for laxative. They use it in all kinds of creams. They use it everywhere, and it's so bad for you. And then, I don't know if it's the Tea Party. Tea Party is this man, the Mad Hatter, that had tea. Not good. And then, if you know the Jobens, right, the people in the last century, so if you know the Jobens, they were polishing those hats with mercury. And that was one of the first time when we noticed this mercury toxicity, because the workers that had Jobens, out, leave it, it's okay. The workers that they had Joben, you know what happened to them? They got dementia and they died, because they were polishing the hats and they were using mercury all the time. And they got dementia, they got crazy, and they died. So then they use it for fur cleaning. They use it for lamps. They use it for all kinds. One time is this boat. They were transporting mercury, and you know what happened to them? They died. And then, guess what? Generation and generation of dentists, they put mercury in people's mouth. Was that good? No. And let's find out why. Now, if you know, this is, do we like the Joben? Yes. But because they used the mercury, they got crazy, just like the Mad Hatter. Do you remember Mad Hatter from the story? You must see it. It's part of the European culture, the Mad Hatter. So they have Mad Hatter disease. So then, in 2002, finally, only in 2002, the US Senate decided the mercury might not be good. And then it was the mercury spelling. And it was, there you go. So what happened to the mercury? Mercury go in the water, and then from the water, it goes in the fish, and then from the fish, we eat it. Also from the water, it goes in the soil. So anything that is onion and garlic or potatoes, they might have mercury. As well, nowadays, they treat industrial chicken with fish compost, and then you get toxicity in the chicken. So then you have a whole generation of people with depression, with sinusitis, with bloatiness that you didn't have before. Our grandparents, they never had depression, did they? No. Or they were suicidal? No. Or they had bloatiness and a whole bunch of people with sinus problems, right? It's from mercury vapors. And then we got a mercury vapor analyzer that what does it do? It verified the mercury. And then one day, it was in the 60s, on the bridge to Minamata, a village, and it was a factory, and they spilled mercury, and the whole village died, and they got these malformations and stuff, entire village. Look how they got, really, and then they protest, and nobody listened to them, but finally they listened to them, they made a monument. That's the Minamata disaster. Something the same happened in Canada, in the American Indian conservation there. I have a friend there, Lynn Katari, she's wonderful. And then they did as well protest that they don't want toxicity from mercury, so the people die. Look how the coin stays on mercury, and we put it in people's mouth. Shame of us. And then here we are. So, symptoms of mercury toxicity. Look at that. If you have 3 of those symptoms, only 3 if you have, you are a victim of mercury toxicity. Now, there are 5 stages. This is my teacher, Hesham El-Esawi. I learned from him. He was in Harley Street. He gave me a job. I was a young dentist. So what we did, we put the patient on sleep. We had all the embassies from the Gulf and everything. And then we put the patients on the sleep. And then we would protect the nose, the throat, the ears. We have vacuums and ventilation. And we remove the mercury, not cutting in the mercury, cutting around the tooth with this small metallic burr that will cut around the tooth, very thin diameter cut. And then with that cut, we remove entire block of mercury. We don't make mercury powder. When I was young, I even had powder of mercury in my underwear when I was working. And from that, maybe we have mercury side effects as a professional disease as dentists. So then after we remove, we test. How do we test? Jerome mercury vapor analyzer. Now, even OMS did not care about mercury, did not care. They let us use it for patients, and students learn, they think it's good, right? Then we have this vapor analyzer and toxicity check. So we got this toxicity check with biomeridian tests. And with that, we see the intolerances, because what happened, as we have this mercury intolerance that attracts another intolerance to chicken, to eggs, to even to fabric. So we have to find out what we are intolerant to in order to remove it, in order to be able to clean the body from mercury. Where we find mercury? Pesticides, fertilizer, laxatives, drinking water, and so on. Even in the thermometer. So, at the dentist, only the thin man, a man of metal, will come and say, "Oh, sir, please give me a mercury filling," right? Human are biological, organic. We want organic, biological materials in us. We don't want to become a radio transistor. We don't. Definitely we don't. This is one article I wrote. You can find it on our EMLA website. And then, where does mercury collect? Because mercury collects in the headache, in the blood, in the nervous system, in the renal system, because that's where it would exhaust, in the colon, and in the articulations. That's where it collects. And then we have the stage. First stage, bloatiness, hair problem, nerve problems, food allergy. We have how you detect it in the feces, in the urine, in the blood, in the sweat. So first grade, cannot sleep, waking tired, loss of concentration, brain fog, mood swings, depression. Second grade, metallic taste, nausea. Third grade, muscle pain, joint pain. But can we remove it? And finally, fifth grade, manic depression, insanity, memory loss, and death. Can we remove the mercury? Yes, we can remove it completely, and the patient feels like flying. So they cause autistic patients, when they give in the vaccine to children, the vaccines had mercury. And you got all this generation of autistic children that again you did not have before. Autistic children. Infantile acrodynia, the pink child, comes from mercury. This is the mercury vapor analyzer, Jerome, and this is biomeridian test. And what we do, we ask the patient not to have these foods. No chicken, eggs, garlic, onion, spring, and leeks, and fish. Do you know the expression drill, fill, bill? Drill the tooth, fill the tooth, bill the tooth. No, we are doctors. We have to think as doctors. Entire body, not just the tooth. We have to think as doctors. That is important. Amalgam fillings are the best. They never fail. They stay there for 50 years. They have good adherence. They have good seal. They have easy placement. They're so strong. It's no better filling than the amalgam filling, but the toxicity is poison, right? And then what happened? You drink hot soup, you drink cold drink, and then the metal contract, expand, and you have a wall between the mercury and the body. And because it contracts and expands, got micro leakage. And then in the micro leakage, you get food entry, bacteria entry, and I never removed a mercury filling in my life not to fight decay under. Every mercury filling I removed that was there for some time had decay under. So if it's not for mercury toxicity, you remove it because of the decay to clean it and to put something that is biocompatible. So then we have the mercury detox. You see the micro leakage. FDA Commission in Hamburg, Spanish dentist, still Henry Schein is the biggest supplier of mercury amalgam in the world. What are we talking about? There are no studies, show no harm. Dental fillings, that is American Dental Association, but now they changed their mind. But it took a long time. So for aesthetic mercury, only thin men would like that. What happened? When the student grind, what happened it is that we have these vapors that show up. So when we have the vapors enter in the nose, they enter in the eyes. For that reason, when we remove the vapors, we have to take them out. So for mercury toxicity, what we want is to have, excuse me, I would like some silence in the room. Thank you very much. If you want to have a conversation, please go out the room. Thank you. What we do, we remove with ultrasonic. We go under the gum. First, we put this protection of the nose, of the throat, of the eyes, of the ear, and we protect ourselves as well. Because if we have this breathing of the vapors, then we are in trouble. So when we remove entire block of mercury. Then we start the detox. We have detox with algae, we have mouthwash, and we have general detox. So from that we get to detox. We continue with the detox we continue until the mercury vapor analyzer finds that is 0 mercury in our body. There you go. So all the spelling we want, we don't want mercury spelling, we want, what do we want? We want just beauty and everything to be biological. And we don't want the Mad Hatter, we want this beautiful hat. You know, the Mad Hatter they were the workers that they were polishing the hats, and then they became mad because of the mercury. That's part of our work, and we love it very much. Now, I want to say a great thank you to our Dr. Raja Johnson, because he organized so beautifully our conference, and to Jacob, Dr. Jacob, and to my old colleague that I admire so much, Dr. Alexander Matthew. A bravo for the beautiful artistic program, scientific program, and as I said, the power, the intellectual power of India is leading the world. And I congratulate you for that. Well done. Now this is me. My last degree is in education from NYU. Before that, I did a PhD and a Master of Science in Biophysics. And I traveled the world. I visited 60 countries, not for holiday. I never had a holiday in my life, but I enjoy so much. So, Einstein made the phenomenon of movement of electrons, right? Stimulation, emission of irradiation. He found the electrons and he found the photon. What is the photon? It's a particle of light. So he said, this particle is emitting and is going to make light, irradiation. And then it took only 60 years until another Jewish boy, Theodor Maiman, made the first machine that used the photon, the first laser. And he put it in the industry to cut. And he cut it metal, he cut it that, and now we cut diamonds. I did cut a diamond myself with a laser neodymium YAG. But only 2 years after, Theodor Maiman, he said, build me one, small one, to use it in oral maxillofacial surgery. His friend in South Africa built him a laser, and he used it in on dogs, and then put it directly in the maxillofacial surgery, and he became the father of laser in medicine. That's me when I was young. I asked him, give me a life advice. And he said, take good care of yourself. And it's true, because if you take good care of yourself, you have a possibility to take good care of the others as well. I also asked him, what do you think of laser hair removal? He said, take a blade. Laser is for serious stuff. Then, 5 years after that, Professor Menster from Hungary, another nice Jewish boy, he said, this laser causes cancer and I'm going to prove it. Laser is really bad and I'm going to prove it. I believe it's so bad and is not going to be used. And he did a double study with mice. One mice irradiated with ruined, and another group of mice not irradiated. And he found out that the blood tests of the mice that were treated with a laser, they were better, and their hair was growing faster. And he discovered photobiomodulation, photoregeneration. He did that, but he didn't know because he was not a researcher. He was just a doctor. For that reason, I advise you, in your work, use 90% dentistry and 10% research and teaching. So you can give back and learn and experiment and make your life in the work more fun. So only 20 years after that, it was one lady, Tina Carew, who said on the herpes lesion, you put the laser on and cause this anti-inflammatory activation, reduce inflammation and regenerate. So there are two things that the photobiomodulation does. Reduce inflammation is one thing and regenerate another one. They're two completely different and they're wonderful. You have reduce inflammation, immediate effect, and regeneration, second effect. Now, what do we have? We have all kinds of lasers invented. Neodymium, CO2, we know them well and they're wonderful. This is spectrum of light. Spectrum of light. So we have all kinds of light. We have visible light that is here, and then photobiomodulation you must learn, because now you're gonna get a certificate that you learn about lasers. So you have to prove. When somebody asks you about lasers, they want to test you if you know, and is only one thing will ask you: what frequency? So the laser infrared, I don't like LED, and I don't like the lasers with light. Use that for disco. I have 8 disco light bulbs. The near infrared is the laser that causes photobiomodulation. And the near infrared starts here, 780, and goes to 1000. So you say, my laser is 830 or 810. When you get a machine, you look directly what frequency. Also, when you use the laser, you have to make sure you have the eyes protected. So in my tests that I did in the clinical trials, we used 2 frequency, 830 nanometer and 630. And what they found out, that the laser works on hemoglobin, melanin, water, scalar. For that reason, the younger people, they have more water. So when we measure the results, they get faster results. But more mature people that have less water, they still have benefit and wonderful results. So what we see on the blood, the action of 830 is not the most stimulation. The most stimulation is at 910. For that reason, now we're making a laser. We make a laser with 910 to treat ambulatory. So instead of having a packet of pills in the pocket full of pills, we have a little light that we can use for regeneration, for anti-inflammatory, for the pills, because we have to change medicine. We have to change. What are all these pills? It's a product. We have to change. Now we have to evolve. We have to evolve. For that reason, the light is really important. Now the tissue penetration. Each laser has different tissue penetration. For that reason, when we have the diode, if you see, it goes all the way to 8 centimeters in the body. For that reason, it can heal heart attack muscle. It can heal the spinal cord. It can go for Alzheimer in the brain. It can do articulation treatment because it's non-invasive. So the light goes and treats 8 centimeters in the body, but depends. We can adapt. So for me, as I was treating the wisdom tooth or the canine, I needed 1 centimeter or 2 centimeters to regenerate the bone of the maxillary. Yes? Or for the sinusitis, 2 centimeters, 3 centimeters. So you put the parameters together. You adjust the parameters. And you don't need too much frequency. This is a photobiomodulation. You don't need strong stuff. You don't need strong. It's 0.5 joules per centimeter. in that place is sort of like an acupuncture without the needles. It's a photobiomodulation that depends on the meridians of the body. That depends on the, because when we do laser on this side, we have to do on the other side. If we have temporomandibular joint on this side, we will apply laser on this side because it goes and the light have effect in the bloodstream and into lymphatic system. For that, when we use the laser for the teeth, we have all kinds of effects, favorable effects in the body. Acne, vaginal candidosis, articulations, and many others. These are all kinds of mechanisms and effects. Anti-inflammatory, tissue repair, analgesia, and what we like it because we are dentists, we like bone regeneration. When we expose with the laser, it can go in short pulse or long exposure can be deeper in the tissue. Now, these are the mechanism of the light with the body. First, the light, it touches the cells, yes? And what happened in the nucleus is oxygen. And this light triggers oxygen, and oxygen goes out and stimulates protein C reactive and open the cell membrane for calcium, potassium pump, natrium, potassium pump, so the oxygen and nutrients function. So if it is a cell that is sick, will regenerate. If it is a cell that is already healthy, will function better. Photobiomodulation does not have side effects. We have to protect our eyes, so when we have the laser, we won't do like this to see if it works. We check with the laser beam on something, and we have a guiding light. Remember, this light is infrared. Infrared is invisible light. As you see, what does it do? It stimulates protein C reactive, pain, anti-inflammatory, tissue repair, and nerve regeneration. So from the tissue to cell to mitochondria to cytochrome to ATP. And the signal of the oxygen from the cell go and does all these clinical benefits. Tissue repair, anti-inflammatory, analgesia, and the effects are local and general. Local and general effects of the light. Even if we apply for the sinusitis, scanning entire area of the sinuses, will have a general effect of anti-inflammatory. So the mechanism, hemostatic effect, pain relief, stimulation, edema, enzymatic activation, protein, cell growth, collagen synthesis. So what is the reaction of the skin with the tissue? It can be that it reacts, the gum, and reflects, or it can be that the laser light is absorbed, or it can be that it just goes through and nothing happened, or it's scattered. For that, in the dental practice, we will not have shiny surfaces. And one pair of glasses with the frequency outside the door with the sign. We have the laser sign, a yellow triangle with a star inside, and then we have those goggles hung down at the door, and inside the practice, every nurse and doctor or a visiting person, they have goggles. The patient also. Now, in the applications of photobiomodulation, we found effects on post-myocardial infarction, on collagen, on the spinal cord, Professor Longo, to regenerate the tissue, on fractures, on all nervous system effects, causing beautiful anti-inflammatory effect in immediate and then post regenerative effect. So if you look for the research that is already there, we have about 27 publications for the last 30 years of my work, and now that I have more time, we publish and publish more, and that's very good. You have to share and you have to publish. Actually, what we say in academia world in New York, we say publish or die. Or we say the students don't take prisoners, they kill you. If you're not a good teacher, right? These are common lasers in dentistry. All kinds, Sirona, and this is the one I used for 30 years, 31 years now, this one here. And if you see, got a screen. Yes, the screen is actually having all the setting. Somebody put all the setting in. 2 millimeter for sinusitis, 1 millimeter for apex of the canine, 1.5 millimeters for wisdom tooth, for gingivitis, 0.5 for wisdom tooth anti-inflammatory, post-alveolitis. So somebody put all the parameters in already. What is that? It becomes artificial intelligence. But artificial intelligence is somebody that put in before, right? So it can be options of parameters. And then it becomes more artificial intelligence. Should we be afraid of it? No. But it's true that if it's in the wrong hands, can do harm, right? But normally you have to think you can always pull the plug on that. So 31% of adults said that would be so nice if my dentist have a laser I like. That means he learn and his technology. But many dentists, they say it's too expensive. I don't have to learn all that. It's not safe. I better use traditional treatments. So what are the benefits in the eyes of the dentist? Increased patient comfort, increased effectiveness of treatment, increased patient acceptance. third home. Fourth thing you do, you buy fourth home. Yes? And then you rent them out. So God will, nothing happen to you ever. But what if something happened to you or to your family? Then you have some income that is there. That's why you study so hard, to protect your family and yourself, to make beautiful life. So, effects on the tissue. All the others I disapprove. But photobiodemulation with infrared light, that's what I like the most. Of course, the others are instruments, mechanisms, tools, because we are engineers as dentists. We are very capable engineers. TCCO2 laser was the first use on medicine. And now you see the medicine is changing from using those torture devices to using the laser to cut tissue at a distance. The medicine is really changing. So we don't touch anymore. So that means sterile treatment, because we don't touch, and it makes a popcorn sound and doesn't touch. So we remove the decay very easy and with very little pain. That is wonderful. So no anesthesia, less bleeding, reduced post-operative edema, early healing, less chance of metastasis. This is my laser. I wanted to bring it, but I left it at the university in Romania, and I couldn't get it back. Actually, this I left in Tanzania at Dar es Salaam University, and this I left in Romania. But if I have laser, I'll bring them to you, and you can use them. So you see the parameters, and we use for photobiomodulation, because we have so much inflammation to be used. This is a Sirona laser that we like very much. That also is in Tanzania at the moment. So we have hard tissue, prevent the caries, detect the caries, prepare, enamel etching, desensitizers. The applications of lasers in dentistry are so much. We have incision, excision, ablation, frenectomy. One application that is really nice in India, they want it, is when you have melanin on the gums and you scrub. Now doing laser for aesthetic is the most boring thing. So for that reason, aesthetic in your practice is very good if you have another room that you can leave it for the boring thing, for teeth whitening and for the aesthetic stuff. If you get a laser to do aesthetic stuff, you must have a nurse that can do it, that is a very patient girl or boy, and you must have an extra room or two. So you have one room that is going to do constantly teeth whitening, because I promise you, you're going to use it, and one room to do constantly aesthetic. Boring. Aesthetic is so boring, and it brings money, so that's good. What other application we can use with dentistry? In fact, all dentistry you can use the laser for. It doesn't replace the classical treatments, but it can help. It can fasten the time of treatment, and it can be very effective. One application that I really like is to, when we have the gum that covers the tooth, to open, to immediate follow gingivectomy, to expose and have no bleeding. For frenectomy, it goes very well, and for all other procedures without bleeding. And you can watch on our website all the videos that we make. You can do that. Another application that is very important, and let's hope that in India they will do for the cancer centers, is treatment with photobiomodulation of oral mucositis and of stomatitis for the cancer patients. It's immediate treatment and is non-invasive, and you can treat it using the laser on the outside of the mouth, because it's infrared, and infrared penetrates the tissues. For periodontal disease, let's say we have bone destruction of 5 millimeters and pockets, and then we do all the state-of-the-art treatments. What do? We clean the tooth. We put the maxillaries in perfect, beautiful occlusion. We check the blood test to make sure that the patient got calcium, got all his biological constants in good shape. No diabetes, no toxicity for liver and kidney, no osteoporosis, and enough calcium and vitamins and stuff. So we make sure that the patient, even if got health problems, become healthy by treatment, general treatment, and locally, no tartar, protocol of hygiene at home with chlorhexidine, propolis be liquid, 1 drop on the gums at night. I promise you it stopped the bleedings in 1 day of the gum. And the patient have to learn and do that protocol 3 times a day. And you say to the patient, I come to your home and verify if you have in your bathroom the chlorhexidine gel and the mouthwash and propolis and the soft toothbrush. And if it's a patient that got really acute inflammation, you ask the patient to take the toothbrush and the toothpaste with them in the pocket. And then you say, if I meet you on the street and I find you without toothbrush in your pocket, you got in trouble. And then you will see, we treated twins. She had a good husband, and they were twins, right? Boy and girl. And he had a wife, and they were drinking, and they were eating McDonald's. And the other couple, I tell them, eat like a goat, only greens, and take a juicer. Citrix and greens, eat like a goat if you want to save your teeth. And then brush gently, put the gel, put the propolis, and go to sleep. You ate outside because you had a conference, you go to the bathroom of the hotel and brush your teeth and rinse. So never ever leave the bacteria to have a party on your gums, not even during the day. When is the most important washing of the teeth? Which is the most important? At night, before we go to sleep. Because if not, you sleep and the bacteria multiply so much, and then you get gingivitis. So we have this study that we did. We separate the patients in 4 subgroups. 2 subgroups, 1 younger, 1 older, and then another 4 healthy patients, patients with diabetes, patients with hepatic and kidney toxicity, and patients with osteoporosis. What do we find after treating them with photobiomodulation? Of course, we did everything in the state-of-the-art of dentistry that we know. We did ultrasonic cleaner. We did the periodontal surgery. We did the perfect bridges and everything to make sure that occlusion is in place and the teeth don't have toxic forces. Everybody got vertical forces, because that is very important. When you have oblique forces, then you got periodontal pockets and you lose the tooth. You must have vertical forces for the tooth to be relaxed. And if the teeth are together united, they don't have too much force on each other. So you must put the teeth in perfect occlusion before treating periodontal disease. Remember, we are not dentists, we are doctors. We are doctors. We think of the patient. Then we do the protocol. This protocol, I learned it from my teacher, Agafita Lefter, the most wonderful teacher, and the most serious and the most severe. Do you think I'm not a severe teacher? I'm the most severe teacher. Like, is crazy severe. Because all my students, they have to have 100, A+. You don't want to be a doctor of 7, you want to be A+, right? Because if you're not a doctor of A+, you're gonna eat the consequences of your mistake in 1 month, in 1 year, in 10 years. So I was explaining to my student at NYU the mistake they did on model. Until I was explaining, until the patient was saying, I do it again, I do it again, I do it again. So to learn is very important as doctors to learn the conscience. The conscience. And you're gonna vaccinate yourself for 2 things, the conscience and never be late. Because if you are late, you lose your life. Who knows what happened in the 10 minutes you are late? Never ever be late. Be like a Swiss watch. You know, in Switzerland, if the train is 5 minutes late, all the watches are outside doing like that. That's what they do in Switzerland. Never ever be late. This is the protocol. What we do, you see, they're not big doses, they're small doses. 0.5, touching interdental space, then scanning entire area.
Excuse me, madam.
Yes.
10 more minutes.
Yes. So this is the tooth, as we do it in the mouth, and then on the outside. Then the bone regeneration, it happens, and then we have the parameters. We measure the parameters and we got the results. In all cases, we found in the benefits of the photobiomodulation. It's very easy and it's cheap, it's new medicine. Did we use other treatments or not? No, we used not much treatments. Bone regeneration happened inside the maxillary, and in all cases we found in the benefits of that. And these are some of, from the clinical cases, some of the beautiful smiles. For example, this lady, she got a chance to find a husband and to find a job before nobody would hire her. He had toxic problems and now he got a beautiful smile. He wanted to get married. So since 1998, NASA decided to give laser to the space, and then also US Army and FDA approved in 2002, and you see all this time passed, we are now in 2024 and still nobody heard about it. Some did. This is a book I wrote in 2007. And let's there be light, and we'll evaluate the devastation of periodontal disease. Thank you very much.
Thank you very much, madam. You took us, gave us an awareness about mercury and took us along the laser. Thank you very much.
Any questions? No questions? I want to invite you to New York for the laser course that we do there. It is a beautiful program. It takes not long, and you can talk with Dr. Raja and Dr. Jacob, and I say namaskaram.
Hello. Okay. The team you're seeing on dais is a wonderful representation from the European Medical Laser Association, and Dr. Dana York is the president of the association, and she wants to seed the educations on laser and ASIC medicine in India. Thank you, ma'am, for flying all the way. Please come on dais for receiving your moment of appreciation. We also have a few words. I know we are closing in onto the valedictory ceremony. 5-minute presentation from the two moderators who are also part of EMLA, and they want to say a few words about the organization. Dr. Dana, you can sit here, and then we'll give the memento. So I invite Dr. Asghar Abbas, ophthalmologist from KIMS, for a few words. So you can start. You can start.
How did I do?
Move it forward. How to move that session.